Okay, so welcome back to this next video in which we are discussing the receptor tyrosine kinases. Okay, right. So in the previous video what we had was a basic introduction to the receptor tyrosine kinases. We saw how they are a family of receptors that is within this bigger superfamily of enzyme-coupled receptors. Okay, we discussed their basic structure, how all of them have their amino terminus of the polypeptide on the extracellular face, and then they have their ligand binding domain, the first major domain that they have after the amino terminus, and then they'll have a membrane spanning alpha helix, and then on their cytoplasmic C-terminal portion they will have a tyrosine kinase domain, which could potentially be split like we've discussed, which is going to fold up into an enzyme which is capable of phosphorylating tyrosine residues within proteins, and then they'll have their C-terminus intracellularly. Okay, so what we now want to discuss is the different families of receptor tyrosine kinases. Okay, so let's go over the page and do this. So, firstly let me tell you about how many different receptor tyrosine kinases there are in humans. So there are 58 different receptor tyrosine kinases. Okay, which isn't that bad compared to the fact that, for instance, there are 800 G protein coupled receptors. So compared to that, it's more comprehensible, 58. Okay, now unfortunately, whereas G protein coupled receptors are grouped into 5 families, receptor tyrosine kinases are grouped into 20 families. Okay, which means that I can't go through every single family of receptor tyrosine kinases, because we'd be here for a very long time going through all 20. So what I'm going to do is I'm going to tell you about the most famous families. Okay, so I'm going to tell you, for instance, about the ErbB family, the platelet-derived growth factor family, the insulin family, the fibroblast growth factor family, the ephrin family, things like that. Okay, right, so I'm going to give you some of the highlights of the 20 families to give you some idea at least. So, the first family of receptor tyrosine kinases that we're going to start off by talking about is the ErbB family. Okay, I probably should put the word family. So this is a subset of the receptor tyrosine kinases. So we've got 58 receptor tyrosine kinases, which themselves are a subset of that great superfamily of enzyme-coupled receptors. And now we're looking at a little one of these 20 families that's within this family of receptor tyrosine kinases, known as the ErbB family. Okay, and this is the family that contains epidermal growth factor receptor, okay, and its analogues. Okay, so let's firstly discuss the structure before we talk about which receptors are actually in the ErbB family. So once again, here is the cell membrane. Okay, now we're going to have our N-terminal up here, extracellularly, as always, and if you don't mind, I'm going to stop putting amino and just abbreviate it to N now. That will become useful when we try to draw insulin receptors later, okay, because it gets really crowded if you try drawing NH2 all the time. Okay, right, so you have important domains within the ligand binding domain of ErbB family members. Okay, so there are 4 separate domains that are usually separated out in the ligand binding domain, so I'll draw all of these out. Okay, so 2 of them are what are known as leucine-rich regions, and then 2 of them are what are known as cysteine-rich regions. Okay, so let me colour these in. So the ones that I'm now colouring in green, so this one next to the amino terminus, and then the one away from that. Okay, so these 2 here. These are what are known as the leucine-rich regions, okay, and the reason they are called the leucine-rich regions is because they contain a lot of leucine residues. So let me just remind you of the structure of the amino acid leucine. Okay, right, so leucine. So once again, I will draw it as an amino acid residue rather than as a pure amino acid, so I'll draw it as though it's within a polypeptide, which is more appropriate considering we're discussing leucines within polypeptides. So here's the amino group as though it's bound to the carboxylic acid group of the previous amino acid. Okay, here's the alpha carbon with a hydrogen coming off. Here's the carboxylic acid group down here. And then again the carboxylic acid group has a bond as though it's bound to the amino group of the next amino acid along. Now the R group of leucine then consists of one carbon here, which has two hydrogens coming off, so a methylene group. Then we have another carbon coming off here, and this carbon is going to have two methyl groups coming off it. One, and then here's the second one here. And then a hydrogen coming off like this. So this is the structure of a leucine amino acid here. Okay, right. So in these leucine-rich regions, these green portions that I've shown here, you're going to have lots of these leucine amino acid residues that I've shown here. And leucine is a good example of a hydrophobic amino acid. Okay, this R group is not very polar at all. You've got bonds between carbon atoms and bonds between carbon atoms and hydrogen atoms. Okay, and the electrons in these bonds are going to be sitting pretty much in the middle between the carbon and the hydrogen, because the pull they're feeling from carbon is roughly the same as the pull they're feeling from hydrogen. So the structure is very, very neutral and therefore doesn't interact well with water. Okay, right. The other two regions, which I'll colour in yellow here, so the ones in between, so it's kind of like a zebra here. So these two yellow regions here, these are known as the cysteine-rich regions. Okay, so these are rich in cysteine amino acids. So once again, I'll show you now the structure of cysteine amino acids. So cysteine has a sulfur atom within it. Okay, so once again, I'll draw you out initially the core amino acid structure. So here's the amino group. Here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group, like so. And now the R group of cysteine consists of a methylene group, like so. And then it's got this special group coming off, which is called a thiol group. Okay, so when you have a sulfur atom bound to a hydrogen atom like that, what I'm circling in yellow here, this is a sulfur atom bound to a hydrogen atom like this. This is called a thiol group. Okay, and it's very similar in chemical properties to an alcohol group. So an alcohol group consists of an oxygen atom with a hydrogen coming off it. So we've just substituted in place of oxygen here, a sulfur atom. Okay, now sulfur has the sulfur is in the same group of the periodic table as oxygen, and therefore has similar chemical properties to oxygen, and therefore thiol groups have similar chemical properties to alcohol groups. Okay, so in these cysteine-rich regions, you're going to have lots of these cysteine residues here. Okay, right, so those are the four main domains which make up the ligand binding domains of these ErbB family receptors. Then, of course, we have the membrane spanning alpha helix, which I'll show in red there, okay, which all of the receptor tyrosine kinases will have. And then you've got your tyrosine kinase domain. Now in the case of the ErbB family members, the tyrosine kinase domain is not a split tyrosine kinase. So the entire thing is all at one sort of region of the primary polypeptide structure. So it's all one portion of the primary polypeptide structure. There's not that kinase insert separating it into two separate regions. Okay, so this is our tyrosine kinase domain. And often people will abbreviate tyrosine kinase domain. Rather than TK, they'll put TYR, short for tyrosine, and then K. So tyrosine kinase domain here. Okay, right. So let's discuss the members of the ErbB family. So there are four members of the ErbB family. There is ErbB1. Okay, now ErbB1 is the most famous. Well, maybe not actually, but ErbB1 is the epidermal growth factor. Okay, receptor rather. Okay, so it's EGFR. And remember, this is short for epidermal growth factor. So EGF stands for epidermal growth factor. It's an important molecule that stimulates cells to divide. Okay, and then the R is for receptor. So the ligand for this receptor tyrosine kinase, ErbB1 or EGFR, is epidermal growth factor. Okay, so epidermal growth factor will bind to its ligand binding domain and activate it. Now, it has another name as well if it needed another one. Okay, its other name is HER1. Okay, and this will become important in a moment. So what does HER1 stand for? Well, HER1 stands for human. Okay, that's the H. The E then stands for epidermal growth factor, which I'll just abbreviate to EGF. And then the R stands for receptor. And then, of course, 1. Okay, so this is the human epidermal growth factor receptor 1. So all of those names you can see for ErbB1 or EGFR or HER1. Okay, right. Now there are other members of the ErbB family as well. There's also ErbB2, ErbB3, ErbB4, and these are also receptors for epidermal growth factor. Okay, so ErbB1, ErbB2, ErbB3, and ErbB4. These are the 4 receptors for epidermal growth factor, and they're all within this ErbB family. Now they also have other names, okay? ErbB2 rather, is also called HER2, so we're using this human epidermal growth factor receptor name, okay? ErbB3 is also called HER3, and ErbB4 is also called HER4. Okay, now HER2 has become quite famous in recent years because it's one of these receptors that is often upregulated in breast cancer. Okay, so cancer cells in breast cancer often have far too high levels of HER2 receptors. Okay, and this seems to be driving their overproliferation. And the very famous drug trastuzumab, also known as Herceptin, so I'll just mention this. It's a monoclonal antibody drug which binds to HER2 receptors. Okay, trastuzumab is its official name. Its brand name is Herceptin. Okay, because it's intercepting HER2 basically, which is why it's called Herceptin. Okay, so it binds to HER2 and stops the epidermal growth factor from being able to bind to HER2, and therefore stops the stimulation of the HER2 receptors, and therefore hopefully reduces the proliferation of the cancerous cells within breast cancer cells which overexpress HER2. Okay, so this is a very important family of receptors both in health and in disease. Okay, so that's the ErbB family, the first family of receptor tyrosine kinases that I want to discuss. The next family that I'd like to discuss is the PDGF family. Okay, another family of receptors which respond to a growth factor. Okay, so this is a running theme that many of the receptor tyrosine kinases are receptors for growth factors. Okay, or rather many of the receptors for growth factors are receptor tyrosine kinases. Okay, so the PDGF family then, which contains the PDGF receptor alpha and the PDGF receptor beta. Okay, so we'll start by having a look at the structure once again, and then we'll talk about the members of this family. Okay, so once again, let me draw the phospholipid bilayer here. Okay, and then once again we'll have our amino terminus extracellularly. Now, this time in PDGF families you have 5 important domains within what we might call the ligand binding domain, which is extracellularly. Okay, so I'll draw these 5 important domains here. So these domains are what are known as immunoglobulin-like domains. Okay, and then I'll draw the transmembrane region. So all 5 of these are the same type of domain here. Okay, they're what are known as immunoglobulin-like domains. Okay, because you find domains like these within antibody molecules, which are also known as immunoglobulin molecules. Okay, so these are domains similar to what you would find in antibodies. So they're called immunoglobulin-like domains. Okay, so I'll colour in these immunoglobulin-like domains in blue here. So 1, 2, 3, 4, 5. So that's the characteristic feature that these platelet-derived growth factor family receptor tyrosine kinases have. They have these 5 immunoglobulin-like domains. And for short, immunoglobulin-like domains are often abbreviated to IG for immunoglobulin. So we abbreviate down our immunoglobulin to IG. And then we maintain the like. So we put IG-like domains. Okay, right. So you have these 5 IG-like domains on the extracellular side of the membrane. Then you have your membrane-spanning alpha helix in red here. Okay, and then on the cytoplasmic side, you then have your tyrosine kinase domain. Now, in the case of the platelet-derived growth factor family, receptor tyrosine kinases, you have a split tyrosine kinase domain. Okay, now I'm going to have to find some different colour to colour it in because I've used blue for the immunoglobulin-like domains. So in turquoise here now, this is the 2 portions, the 2 regions of the linear polypeptide that are going to bind together to make the full tyrosine kinase enzyme. Okay, and by the way, at the moment we're just discussing the inactive enzyme. We're not discussing the active enzyme yet. We'll talk about active enzymes later and how we activate them. We're just talking about the inactive enzyme sitting within the phospholipid bilayer at the moment. Okay, right. So here we have our split tyrosine kinase domain. Okay, right. And that portion in between the 2 halves of the split tyrosine kinase domain is then called the kinase insert. And I'll colour in the kinase insert in a moment in, I think, vivid purple. Okay, so this is the basic structure then of all of the platelet-derived growth factor family receptor tyrosine kinases. Now, there are many members of this family, but I'm just going to name a few. I'm just going to name the obvious ones, which are the platelet-derived growth factor receptors. Okay, so the 2 platelet-derived growth factor receptors, the platelet-derived growth factor receptor alpha, and also the platelet-derived growth factor receptor beta, are both receptor tyrosine kinases within this platelet-derived growth factor family of receptor tyrosine kinases. So I think I need at least once to write out the full name for PDGF. So it is, the P is for platelet, the D is for derived, and then GF, once again, is for growth factor. So this is the full name for PDGF, platelet-derived growth factor. Okay, now underline it in R. So here we go. This is PDGF. Right, so that's the next major family of receptor tyrosine kinases. Okay, the PDGF family of receptor tyrosine kinases. The next family I want to turn my attention to is the VEGF family of receptor tyrosine kinases. Okay, and this is very, very similar to the platelet-derived growth factor family. Okay, so I'll try and squeeze this in here because it would be nice to have them on the same page so that we can compare them. So another family that's named after a growth factor, basically, and this is because the main members of this family are once again going to be receptors for this growth factor. Okay, vascular endothelial growth factor. So let's draw its structure here again. Okay. So here is the phospholipid bilayer. Once again, we have the N-terminus extracellularly. This time, we again have an extracellular ligand binding domain, which consists of a huge number of repeated immunoglobulin-like domains. So we have lots of immunoglobulin domains, and it's even more than in the case of the platelet-derived growth factor family of receptor tyrosine kinases this time. Okay, so usually you have around 7 immunoglobulin-like domains in VEGF family receptor tyrosine kinases. So all of these are immunoglobulin-like domains, and I'll abbreviate immunoglobulin-like domain to IG-like domain. Okay, so once again, I'll colour all of these 7 immunoglobulin domains in blue. 1, 2, 3, 4, 5, 6, 7. Okay, so these are all immunoglobulin-like domains in blue here. Okay, then we have a membrane-spanning alpha helix here that straddles the membrane. We'll have that in red. And then on the cytoplasmic side, once again we have a split tyrosine kinase domain in vascular endothelial growth factor family receptor tyrosine kinases. Okay, so here are the two halves of the tyrosine kinase domain, one here and one here. And then that portion in the middle, once again, is a kinase insert, which I'll colour in purple here. So this is the structure of all VEGF family receptor tyrosine kinases. Okay, so within this family there are three main receptors that I want to talk about, which are the vascular endothelial growth factor receptors. So there are three of these. The vascular endothelial growth factor receptor 1, the vascular endothelial growth factor receptor 2, and the vascular endothelial growth factor receptor 3. And once again, their ligand is the vascular endothelial growth factor, or VEGF. So the V is for vascular, the E is then for endothelial, okay, and then the GF is for growth factor. Okay, so there are different forms of vascular endothelial growth factor, which will have different specificities for the different types of vascular endothelial growth factor receptor. But we'll keep this nice and general and just talk about vascular endothelial growth factor, VEGF. Okay, right. So that's the next family then of receptor tyrosine kinases, the vascular endothelial growth factor family. So we've now seen three families of receptor tyrosine kinases, which are all receptors for growth factors, and they're all named after the fact that their leading members are receptors for growth factors. Okay, that's going to continue. We're now going to move on to the fibroblast growth factor family, FGF family. Okay, so the next family then. Okay, again, this is going to have a sort of similar structure to the PDGF family that we've seen. It's going to have immunoglobulin-like domains within it. So, once again, I'll draw the phospholipid bilayer here, and I'll try and draw these smaller now so that we can save a bit of paper. Okay, so the amino terminus will be extracellularly. Okay, then we're going to have three immunoglobulin-like domains in the case of FGF family receptor tyrosine kinases, but we're also going to have a structure in between two of them. Okay, so let me show you this. So we'll have our amino terminus here, and then we've got our first immunoglobulin-like domain here. We've then got a special structure next up known as the acid box, and I'll come back to this in a moment. Then we've got two more immunoglobulin-like domains, one here and one here, before we then have the transmembrane alpha helix. Okay, so let me colour the immunoglobulin-like domains in blue here then. We've got these three immunoglobulin-like domains. So this one, and this one. All of these in blue. These are immunoglobulin-like domains, Ig-like domains. Okay, similar to these structures that we have within antibodies. Okay, then, that region that you've got in between the first and the second immunoglobulin-like domain that I've now coloured in red here, this structure is what's known as, so I'll just straighten this up a little bit. This structure is what is known as the acid box. Okay, and it's called that because it's going to have a large number of residues within it that are acidic. Okay, so let me give you an example of an acidic residue. So the sort of archetypal example of an acidic residue is an aspartic acid residue. Okay, so once again I'll draw the core amino acid structure firstly. So here's the amino group, and again I'm drawing it as though it's a residue. Here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group, again as though it's bound to the amino group of the next amino acid along. Okay, and then the R group of aspartic acid consists of a methylene group, then with a carboxylic acid group coming off here. Okay, and this then is the R group of aspartic acid. Okay, this 2-carbon carboxylic acid side chain here. This is the R group of aspartic acid. Now, it's an acid, because it can donate this proton off this alcohol group on this carboxylic acid group away into the solution. Okay, so if we draw this bond in a little bit more detail, here's the oxygen, and it's bound by a single covalent bond to this hydrogen. Okay, so there'll be 2 electrons in that bond, one from the oxygen, so we'll colour up in the one from the oxygen here in red, so this is the oxygen's electron here, and then in blue here, this is the hydrogen, and we'll have that as the hydrogen's electron. Okay, so there are 2 electrons in this bond, one from the oxygen, one from the hydrogen. Okay, but what can happen is that the hydrogen can leave this bond and leave its electron behind with the oxygen. Okay, now what is left of a hydrogen atom if you leave the electron behind with the oxygen atom? Well, most hydrogen atoms only consist of a proton at the center within the nucleus, and then with an electron orbiting around the outside. Okay, so we'll have the electron here in blue and the proton here in turquoise. Okay, this is the most common isotope of hydrogen, okay, called protium, where you have absolutely no neutrons. There are other isotopes of hydrogen where you do have neutrons within the nucleus. For instance, deuterium refers to hydrogen where you have a single neutron in the nucleus as well. Okay, so one nucleus is then deuterium, so this one's deuterium. One neutron is deuterium, not nucleus. Okay, and then finally you've also got tritium, which then is 2 neutrons in the nucleus. So we'll get an additional one here, which I'll colour in orange. Okay, right. But those isotopes of hydrogen are much, much rarer than protium. Protium is the main form of hydrogen, okay, and it just consists of a proton and an electron. So usually, when this hydrogen leaves its electron with this oxygen, what will be happening is you're just giving away a single proton, and when you give away a single proton into solution, that's the definition of an acid. So aspartic acid is therefore an acid. Okay, so this acid box here is going to have a huge number of residues in, a higher than normal number of residues which are acidic and therefore can donate protons away into solution. Now, what's the significance of this? Well, what happens once the residue has actually donated its proton away into solutions? Let's imagine that the proton's gone. The oxygen is left with a negative charge. Now, the name for aspartic acid, once it has lost its proton, once it's donated its proton away into solution, is aspartate. Okay, so aspartic acid, strictly speaking, is only the name for this molecule that I've drawn here. Okay, when it's got that hydrogen attached, once it's donated its proton away, and you've got a negative charge on this oxygen, but no hydrogen anymore. Okay, that isn't called aspartic acid anymore, that's called aspartate. Okay, and the fancy word for this is that it's the conjugate, well, the fancy term at least for this is that it's the conjugate base of aspartic acid. Okay, so a conjugate base is a general term for acids, okay? Well, for the molecule that you get from an acid once the acid has donated its proton away. So it's a running theme. It doesn't just apply to aspartic acid. You can take any old acid you like, and once the acid molecule has donated away its proton, it's no longer capable of donating another proton, okay? So the molecule that you've got left over after you've donated your proton away, okay, that can't donate another proton away, and therefore it's no longer an acid, because the definition of an acid molecule is a molecule that's capable of donating a proton away into solution. The molecule that you're talking about has already donated its proton away into solution, and it can't therefore donate another one. So it's no longer an acid. Instead, it's actually a base, because rather than donating a proton away in solution, it's actually able to receive a proton from solution, which is the definition of a base. Okay, so because you've got the negative charge, for instance, on this oxygen here, you can accept a proton from solution, and therefore you are a base. So aspartate is the conjugate base of aspartic acid. So, what was the aim of telling you all of this? Well, when these acidic residues donate their protons away into solution, which at any moment in time, some of the residues within this acid box will have donated their protons away, and others will still have it bound to them. Okay, so when some of them have donated their protons away, those that have donated their protons away will have a negative charge on them. So basically, regions where you have acidic boxes within your polypeptide structure tend to have also negative charge on them, because some of the residues will be existing in the conjugate base form, and these will have a negative charge, whereas the ones which are in the original acid form, they will just be neutral. So regions of polypeptides where you have a high proportion of acidic residues tend to also carry a negative charge. Okay, so that's that acid box then. Then what we have is the membrane spanning alpha helix, which I'll colour in red again. And then we have, again, a split tyrosine kinase domain. So the tyrosine kinase enzyme is made up from two portions of the polypeptide that are not next to each other, basically. Okay, there's an insert between the two portions of the linear polypeptide which actually make up the tyrosine kinase enzyme. Okay, so these two portions in turquoise, these are the two halves of the split tyrosine kinase domain, and that portion in between then is the kinase insert. Okay, so this is the fibroblast growth factor family of receptor tyrosine kinases. So what receptors are in here? Well, surprise, surprise, it's the fibroblast growth factor receptors. Okay, so there are four of these. There's the fibroblast growth factor receptor 1, the fibroblast growth factor receptor 2, the fibroblast growth factor receptor 3, and then finally the fibroblast growth factor receptor 4. And once again, I feel as though I should at least once write out fibroblast growth factor in full. So FGF stands for fibroblast growth factor, which is the ligand, and we're talking about the receptor then for fibroblast growth factor. So we're talking about the fibroblast growth factor receptor, or the FGFR here. Okay, right. So we'll call it there for this video, and in the next video what we'll do is continue talking about receptor tyrosine kinase families. We've now been through 4. I'm going to show you 3 more. Okay, firstly I'm going to show you the TRK family, the TRK family, and I'm also then going to show you the ETH family, the EPH family. Okay, then what we're going to discuss is we're going to discuss the activation of these 6 families of receptor tyrosine kinases that I've talked about, and then we're going to discuss the family that's very famous but which is the odd one out, basically. It's different from all the rest, which is the insulin family of receptors, which contains the insulin receptor, potentially one of the most famous receptor tyrosine kinases. Okay, we'll talk about that receptor and then how it's activated, and specifically we'll look at how its activation and its structure is slightly different from the other receptor tyrosine kinases which we've discussed so far.